Hey, Bio 1 students, let's wrap up notes here. This will be the last couple slides. We'll have a test coming up here in a couple days. So let's go through the microscope. We're actually going to do a microscope lab if we have time, and you'll learn how to use one. Many of you that have taken bio before, if you're not in Bio 1 honors, you may have taken environmental science and used a microscope before. But there are basically a couple different types of microscopes. The one we use is called a compound light microscope, or an LM. It uses a series of lenses and ordinary light to magnify an image. The problem with compound light microscopes is they have some limitations because of these two concepts on the screen, resolution and magnification. The problem is that the more you enlarge an object, okay, so magnification is simply, as you can see here, it's just the ability of a microscope to make an image bigger. Resolution's how good does it produce a clear image. The problem is if I make these slides so big that I lose my resolution, they don't do you any good. So if you get things beyond 2000x, you're not going to get a clear image. So we're kind of limited. You really can't buy a compound microscope that has like a 5000 magnification. Not that we can't make lenses to make things that big. The problem is you won't be able to see what you're looking at, so it's kind of worthless. So that's kind of limitations with an LM, but they're reasonable to buy. We have a lot of them here at school. They do what we need them to do. You're just not going to see huge magnification powers. Good resolution, not good magnification power. So we are kind of limited that way, but we can look at living things and do some stuff like that. Now, another type of microscope would be electrons. Electrons don't use light. You don't look through them like we do our microscopes. They don't magnify like that. You actually are shooting a beam of electrons down through a thinly sliced specimen. The problem is we're using magnetic lenses and shooting electrons through a specimen. You can't view living things. That's the downside. But you can get magnification powers up to 200,000x, which is just amazing. A lot of your pictures in your textbooks and on the internet and things you see out there, they're made with these types of microscopes. Scanning is the same type of microscope, except instead of beaming them through, we bounce them off the surface. They're not sliced or sprayed with a metal coating, same problem. You can't view a living thing. But man, can you get some great pictures with SEMs, okay? You have a fluorescent screen, and you get these really nice pictures of different types of organisms. I'll show you here in a minute. These are great microscopes. The downside to them, to make slides, it takes forever when we use electron microscopes. Now, these were old ones. When I was in college, we didn't like using them because it took us an hour to make one slide. So that's kind of the downside to them, but they produce some cool looking pictures. You can see the price tag on some of these microscopes. This is the Quantum. This is really a sophisticated microscope. Yeah, I don't think we can spend $100,000 on one. But you can see actually what an atom kind of looks like, the evidence of an atom. This is what TEMs looked like back in the 80s. They took up a whole room. Then our technology got better. In the 90s, they got a little smaller, and now we got them desktop. Okay, so expensive machines. They're not cheap. So you may see one in college, but you're not going to see one in a high school setting. But these are some, let's go back a little bit, some great pictures that are produced by these crazy things. Look at these pictures. This is actually what toilet paper looks like up close, kind of cool. Bacteria, mitochondria, and stuff like that. Unbelievable pictures, okay? SEMs, same deal. They used to be huge rooms, then they started to get a little bit better, but they're still big machines hooked up to a lot of technology. But you get some of the craziest pictures with them. This is a staple going through a piece of paper. That black stuff's bacteria on the staple, so don't jam a staple into your arm. It's got a lot of bacteria on it. And look how the paper explodes. Kind of crazy, okay? This is actually the backside of a flea, if you can believe that. Fleas are hard enough to see, and then you magnify and see the backside of them. snowflakes, kind of appropriate for coming up this time of year, gills of a fish, just some crazy, crazy pictures we get with these electron microscopes. This is a spider up close. You can actually see its eyes and its antenna and things like that. So you get some really crazy pictures. This one's kind of, if you're arachnophobe, sorry, but you just get some very crisp, good-looking images. We're going to use the microscope a lot this year. We will not use these microscopes, obviously, because they are so expensive to buy, and we just can't afford to do that at a high school setting. But we will use the light microscope a lot. Okay, so that's it for notes. We'll do a review game coming up here. I'll post notes online, and then we'll take a test over this chapter. So look through your notes. Everything that's highlighted in red would be good to study. That's good test questions. All my tests come from the notes. So hopefully you've been taking good notes, and we'll be ready for the test coming up here. So, hey, have a good day. We'll see you later. Bye.